Morning, Dave. Morning, everyone. So, hello. On behalf of my co-chair, Dulce, and the two judges, Tony Moore and Julie Daniels, I'd like to welcome everyone to the Early Career Rapid Fire session. You're all most welcome. So, these are brief scientific talks of 4 minutes with 2 minutes for questioning from the judges directly afterwards. And these are aimed at a scientific audience, but you will find lay summaries in the abstract booklet from page 48 onwards. And also, I'm sure the speakers would be happy to chat you through any parts or expand on any bits you'd like to hear about in the Zoom networking room, either perhaps in the break or directly after the prize announcement. So the presenters are all at early stages of their career, perhaps at the doctoral or postdoctoral stage. They have not yet established their own research group. And they are from a variety of backgrounds, some medical and some purely scientific, but all of them are doing Aniridia research or research very relevant to Aniridia. So I'd like to introduce the panel you see in front of you. So my name's Nikki Hall. I'm a registrar, an ophthalmology doctor in Edinburgh. I do Aniridia genetics research in David Fitzpatrick's group at the MRC Human Genetics Unit. Dulce is my co-chair, and Dulce is a postdoctoral researcher in Maria Musaji's group in London. And then we have Tony Moore, who is Professor Emeritus of Ophthalmology and has worked in London and San Francisco, amongst perhaps other places as well during his career. And then we have Professor Julie Daniels, whose corneal work you heard about yesterday, and who is also a professor in London. So there are going to be two prizes today. There is a £100 judges prize, which will be decided by Julie and Tony. And then there will also be a £50 popular vote prize. Now, in order to vote for that, you need to use the poll function on the live conference chat. So you may see in the chat next to the main screen, there is a second little tab next to the chat that says polls, and the poll is or will be set up on there in order for you to vote for your favorite talk towards the end of the session. The two prizes cannot be won by the same person. The £100 judges prize has been very kindly donated by Veronica van Hanegem, and the £50 popular vote prize is very kindly donated by Aniridia Network. So without further ado, I am going to hand over to my co-chair Dulce to introduce the first speaker.
Good morning, everyone. So let's start. The first speaker today is Alexandra Filatova. She's a research scientist in Functional Genomics Lab of the Research Center for Medical Genetics in Moscow in Russia. And her talk will be Functional Analysis of PAX6 Non-Coding Nucleotide Variants Revealed a New Molecular Mechanism of Congenital Aniridia Pathogenesis.
Hello, dear colleagues. My name is Alexandra Filatova, and I am a researcher from the Research Center for Medical Genetics, Moscow, Russia. A previously conducted molecular genetic study of a large cohort of Russian patients with congenital aniridia revealed two groups of PAX6 nucleotide variants, which pathogenicity was not obvious and required an experimental investigation. The first group includes non-canonical splice variants. We found 8 such variants in Russian patients, 6 intronic and 2 exonic, including 1 missense and 1 synonymous variant. To analyze these variants, we created 4 mini-gene constructions and performed a mini-gene assay. Our results demonstrate that 7 out of 8 studied variants, including missense variant and synonymous variant, disrupt normal splicing. It leads to the PAX6 frame shift, followed by an activation of nonsense-mediated mRNA decay. Thus, we show that studied non-canonical splice variants are actually loss-of-function variants, and they lead to the development of the congenital aniridia. The second interesting group of PAX6 non-coding variants are variants located in the 5' untranslated region. We found 4 such variants in Russian patients. Additionally, 14 variants were also reported in the literature, but the mechanism of their pathogenicity remained unclear. We performed a functional study for 4 variants that we identified in Russian patients and 1 previously reported variant. For this, we cloned a full-length PAX6 5' UTR, including full-size introns, into a special vector and fused this 5' UTR with luciferase ORF. Using site-directed mutagenesis, we introduced studied variants, and after this, all obtained plasmids were transfected into 2 cell lines. At first, we performed RT-PCR of transfected cells to evaluate the possible effect of variants on splicing. We found that 2 intronic variants led to an exon 3 skipping, while 3 exonic variants did not affect splicing. Next, we performed a luciferase assay in tandem with qPCR and found that all 5' UTR variants led to a depletion of protein translation of protein level, while mRNA expression level was not significantly reduced. Thus, we conclude that studied variants decrease the protein translation efficiency. To understand the underlying mechanism of how variants influence on translation efficiency, we performed a bioinformatic analysis of available RiboSeq data, and we predicted the presence of small upstream ORF in the PAX6 5' UTR. Moreover, we revealed that all studied PAX6 5' UTR variants might disrupt this upstream ORF in the same manner. Splice variants, as well as small indel variants, lead to the same frame shift and formation of extended ORF, and this extended ORF overlaps with the downstream PAX6 coding sequence and thereby may reduce its translation efficiency. To validate this hypothesis, we performed an additional mutagenesis experiment. We disrupt upstream ORF translation by start codon removing or by stop codon insertion. And when we performed this in wild-type context, we did not observe any changes in translation efficiency. While when we disrupt upstream ORF in context of patient-derived variant, we observed a full rescue of protein translation efficiency. To summarize, we discovered a new molecular mechanism of congenital aniridia pathogenesis through upstream ORF frame shift. Moreover, we conclude that at least 15 out of 18 known PAX6 5' UTR variants realize their pathogenicity mainly through this mechanism. And I want to thank my colleagues who participated in this work, and thank you for your attention.
Thank you for a very clear talk. It was very, very interesting. So what proportion of your patients with aniridia had these unusual mutations?
Hello, thank you for your question. Mutations in 5' UTR, it was approximately 4 different mutations in 4 different patients in our cohort. So it's 3% of all variants in PAX6 that we obtained.
Right, and were they all sporadic patients, or could you show segregation in families?
3 out of these patients, it was de novo variants, and 1 is segregated in family, if it's correctly.
Yeah. Are there any more questions online? No. Well, thank you very much. A very, very interesting talk. Thank you.
Good morning. My name is Philippa Harding. I'm a PhD student in Maria Musaji's group at the UCL Institute of Ophthalmology. Today I would like to show you some of our work investigating PAX6 associated eye conditions, aniridia and microphthalmia using cellular models. For the eye to form properly during development, a specific set of genes have to be switched on or expressed at the right time in the right place. If a gene is not switched on or off at the right time, it can prevent correct movements and changes in cells which form the eye. Changes in DNA can prevent genes from being switched on, leading to eye conditions. One gene which is very important for early eye development is PAX6. PAX6 is a transcription factor, meaning it helps control eye development by ensuring the correct genes are switched on at the right times. Because PAX6 regulates so many genes in eye development, changes in PAX6 DNA can result in a range of ocular disorders, including aniridia, where the iris does not form properly, and further problems like cataracts often occur. Genes are made up of chains of molecules. If one of the molecules in a sequence is swapped, it can prevent the gene from working, which we call a missense variant. If a missense variant occurs in a specific part of PAX6 which is important for it to function, it leads to severe eye conditions, including microphthalmia, where patients are born with an eye which is small and underdeveloped. How different types of genetic changes in PAX6 cause different eye conditions is still unclear, because the precise way PAX6 switches genes on and off in eye development is not well understood. In this study, we aimed to investigate PAX6 using stem cell models. Pluripotent stem cells are a special kind of cell which can form any cell type in the body by switching on the right combination of genes. We can make induced pluripotent stem cells, or iPSCs, from adult skin cells. To do this, we take a small biopsy of skin from a patient. From this skin biopsy, we grow their skin cells and add some growth factors with a burst of electricity to induce the skin cells to form stem cells. By providing the stem cells with certain nutrients, we can encourage them to turn on genes needed to form an early eye, creating a model that we can study to find out more about eye development. We can make these models from patients, where the cells contain the DNA change which causes their eye condition. By looking at cells from patients with a missense change in PAX6, causing microphthalmia and aniridia, as well as cells from a patient with only aniridia, we can understand how different PAX6 changes cause different eye conditions. What we found was that many genes needed for early eye development were not switched on in the cells from the patient with aniridia and microphthalmia. Fluorescent staining of the cells showed that another important gene, VSX2, shown in red, was missing in the cells from the microphthalmia with aniridia patient, but not the cells from the patient who only had aniridia. This showed us that the particular missense DNA change in the microphthalmia patient was preventing VSX2 from being turned on. To find out which other genes might be causing microphthalmia and aniridia, we sent the cells for sequencing of all the genes which were switched on, a technique called RNA sequencing, and we are now analysing this data. In conclusion, we made a cell model of eye development for a patient with aniridia and microphthalmia and a patient with only aniridia to study PAX6 function. Important genes were switched off in patient cells, which can tell us about PAX6 function in eye development and how DNA changes in PAX6 cause aniridia and microphthalmia. I would like to thank the whole Musaji team for their work, our funders, Moorfields Eye Charity and Wellcome, as well as patients who donated skin biopsies, without whom this project would not be possible. Finally, I'd like to thank you all for listening, and please follow our research on Facebook and Twitter.
Well, thank you again for a very clear and interesting talk. One of the it's very difficult in a very short talk to get the across, but how far do you develop the cells down the line before you start looking at downstream effects? And what do they look like? Do they look like? Are there differences in morphology between the cells with the mutations and normal individuals, cells of normal individuals?
Yeah, that's a great question. So we tend to look at the cells after about 20 days in the cell culture, and then also at 35 days. So at 20 days, we think they represent the kind of vesicle stage of development, sort of like just after the eye field has formed and they're starting to look at like the early eyes. And then at day 35, they start to sort of form two different layers of cells, which is like the neural retina and the retinal pigmented epithelium kind of separate out. And we definitely see that in the patient cells, particularly the microphthalmia patient cells, there was a much lower efficiency of forming those structures. So when you look at them just down a normal microscope, you can see sort of like a bright tissue, and there's much fewer of those in the cells from the patients. and they tend to sort of have not quite as they're not as thick, and they're not quite as like stratified, like lots of layers of cells. So yeah, you can definitely see some like morphological differences quite early.
Yeah, and you don't know whether you can grow these cells out into a a layer of pigment epithelium or rudimentary photoreceptor cells.
So I haven't tried with these cells. So the latest I took them was up to 50 days, where you start to see sort of photoreceptor markers that they're sort of differentiating further. And I know other people, including some people in our lab, have tried taking them sort of like further down those lines. I've tried a different protocol, which is specifically differentiating them into the retinal pigmented epithelium, and that worked fairly well because you saw very distinct differences in the sort of structure and the ability of them to pigment from the PAX6 cells compared to the wild type cells. So yeah, definitely like options.
And there's one quick question, if you could answer it very briefly, is do you think how easy would it be in these cell systems to use CRISPR to correct a mutation?
Again, I have tried this. I didn't find it particularly easy because stem cells are sort of notoriously bad at taking up the CRISPR, but it's definitely possible, and other people have successfully managed to do it. And again, other people in our lab are trying to do it with the with different cell types and stuff. So there's sort of different methods whether it's better to maybe transfect the skin cells before you transfect them into the IPS might be more successful. But yeah, I haven't spent lots of time doing it, but it's possible. It's tricky.
Yeah. Good. Thank you very much. We have to move on. Thanks for a great talk.
Sorry. Apologies before that I couldn't introduce Philippa, but she did that very well herself. Julie is having a bit of issues logging in, so Tony will be doing most of the questions for now until we sort this. So shall I introduce the next one then? The next talk will be by Hilde Pedersen. She is a postdoctoral researcher at the University of Southeastern Norway, and her talk is called Retinal Structure and Genotype in Congenital Aniridia.
Hello everyone, I'm Hilde R. Pedersen from the National Center for Optics, Vision and Eye Care at the University of Southeastern Norway. First of all, I would like to thank the organizers for giving me the opportunity to present our study about the retinal structure and genotype in congenital aniridia. I also want to acknowledge all my collaborators. The majority of persons with aniridia have foveal hypoplasia, which may be the main limiting factor in the visual function one can achieve. The purpose of this study was to investigate thickness of the retinal layers and its association with visual function and PAX6 genotype. 37 persons with congenital aniridia and 58 normal controls were included. To investigate retinal structure, we used optical coherence tomography to obtain cross-sectional images of the macular area. This graph shows the variation in thickness of outer retinal layers comprised of the retinal pigment epithelium, the photoreceptor outer and inner segments, and the outer nuclear layer, marked with yellow in the OCT images to the left. The normal thickness of the outer retinal layers is shown by the black solid line and peaks at the foveal center, which reflects the central packing and elongation of the foveal cones. The line with blue squares and dashed lines represent the mean ±1 standard deviation for the participants with aniridia, and shows that the outer retinal layers were significantly thinner in aniridia across the whole horizontal meridian compared with normal controls, and particularly in the foveal center. This suggests that cone specialization and migration has not occurred to a full extent, resulting in immature foveal cones and decreased cone density. The reduced thickness of the outer retinal layers were correlated strongly with poor visual acuity, and was the strongest predictor of high contrast visual acuity, accounting for approximately 66% of variation in visual acuity. We also compared different PAX6 genotypes with structural changes in the retina in persons with aniridia. Mutations in PAX6 or regulatory regions were found in 97% of the participants, and all participants with mutations in PAX6 also had foveal hypoplasia. On the left you can see a schematic presentation of the PAX6 gene, and the box plot to the right shows the differences in foveal outer retinal layer thickness between participants who have mutations within coding, non-coding or flanking regulatory regions of the PAX6 gene. Those with mutations in the protein coding regions of the PAX6 gene had generally thinner outer retinal layers, while participants with mutations in the untranslated region 5' of PAX6 on average had better developed foveals, seen as thicker outer retinal layers, than those with mutations in the coding regions of the PAX6 gene. The variants associated with the thickest foveal outer retinal layers and also the mildest retinal phenotypes were mutations outside the PAX6 gene that left the PAX6 transcriptional region intact, even though these participants had complete iris hypoplasia. So in conclusion, despite the large phenotypic variation, detailed genotyping and analysis of the retinal layers show that the position and extent of the PAX6 mutation may be predictive for the severity of foveal underdevelopment. Such knowledge is of importance in the clinical management of persons with congenital aniridia to understand limits and potential related to visual function. Thank you for your attention, and I'll be happy to answer any questions.
Thank you. Thank you again for a very interesting and clear talk. You didn't mention nystagmus at all. I mean, it's very difficult to talk. So presumably your controls did not have nystagmus, and I just wondered how you got around the problem of eye movement artifacts.
Yeah, that was actually a problem because a lot of them had nystagmus, of course. So with OCT imaging, we use a lot of time because when the eye was moving, of course, it took longer time when we were going to average several frames. But we also adapted the imaging so that they could sit with their eye or with their head in position with where the nystagmus had the least amplitude. So yeah.
You managed to get around the problem. Yeah. And the other question I had was when you nicely correlated the outer retinal changes with the type of mutation, did the persistence of the inner retinal layers at the fovea correlate very well with the outer retinal changes, or were they and with the mutation, or did that not correlate well?
No, not necessarily, because most of the patients with the foveal hypoplasia, of course, have at least in the center, have thicker inner retinal layers, or they are present. So we saw that the outer retinal layers could have been developed a bit despite the inner retinal layers present. So of course that's the different stages of hypoplasia as well, but also within the different stages of hypoplasia we saw differences in the outer retinal layer thickness. Yeah.
Good. There's a question from Julie Daniels about how these phenotypic differences might impact any future therapy.
What we since we saw that the retina has developed in some patients with aniridia, because we know that the outer retinal layers is not developing necessarily before after birth. That could be, of course, a target to early intervention, if it's gene therapy or if it's a kind of other visual stimulation or anything. There needs to be more studies to figure that out, of course.
Yeah, thank you very much. And I apologize to members of the audience that posted some questions for you. We haven't time to consider them, but thank you again.
Thank you.
You can keep posting your questions, though. We'll try to answer them afterwards in the Zoom meeting where the speakers will be available. So next we'll have Vivian Kidd. She's an ophthalmology trainee and clinical research fellow. She works at the Moorfields Eye Hospital with Maria Mukherjee in London. Her talk will be called Longitudinal Genotype-Phenotype Analysis of 86 Patients with Molecular Confirmed PAX6 Mutations.
Kit, and I'm presenting longitudinal genotype-phenotype analysis of 86 aniridia patients with molecularly confirmed PAX6 mutations. So aniridia is caused by a mutation in the PAX6 gene. Clinically, patients with aniridia can present with varying levels of underdevelopment of the iris, known as iris hyperplasia, nystagmus, aniridia-related keratopathy, which affects the cornea, cataracts, glaucoma, and underdevelopment of the part of the eye involved with central vision, known as foveal hyperplasia. What we set out to do with our research was to identify if genotype-phenotype correlations exist. This means we wanted to identify if the type of mutation in the PAX6 gene can affect how a patient presents clinically. We identified 86 patients from 62 families at Moorfields Eye Hospital with a known diagnosis of aniridia and a molecularly confirmed mutation in the PAX6 gene. These patients were then divided into various groups based on their mutation type. A retrospective review was then performed from their first clinical visit to their most recent clinical visit. We looked at several parameters, which I'll discuss in the upcoming slides. So one of the parameters was visual acuity. If we look to the middle, we demonstrated that patients with no surgical intervention maintained better visual acuity compared to those with surgical intervention. Surgery included procedures for glaucoma, cataracts, and aniridia-related keratopathy. Amongst all patients which had no surgery, so surgically naive, and amongst those with surgical intervention, patients from the missense mutation group, highlighted by the line in red, maintained the best visual acuity amongst all groups. Another parameter we looked at was iris architecture. So we looked to the left. Amongst all mutation groups, patients from the missense mutation group were more likely to have a full, complete iris. Comparatively, patients from the nonsense, intronic, and gene deletion groups were more likely to have complete iris hyperplasia. On the right of the image, we demonstrate the variability amongst the mutation groups at the age of which cataracts and glaucoma was diagnosed. So in conclusion, to the best of our knowledge, this is the largest in-depth clinical description of PAX6 genotype-phenotype correlations and natural history of aniridia in the United Kingdom to date. In our study, the missense mutation group demonstrated better long-term visual acuity, a lower prevalence of aniridia keratopathy, which affects the cornea, and milder grades of underdevelopment of the iris and the fovea, which affects central vision. Now, there is, however, a caveat to this. There has been publications in the literature which has demonstrated missense mutations and patients presenting with more severe phenotypes, in particular, severe microphthalmia. The take-home message from our presentation is that when faced with a patient with aniridia, first and foremost, we should perform genetic testing to rule out Wagner syndrome. But by facilitating better understanding of genotype-phenotype correlations, we may be able to better understand aniridia, but also better facilitate clinical outcome measures. I'd like to take this opportunity to thank you all for listening, but also to my co-authors, Dr. Dalpsley Makunya, Dr. Ahmed Hagag, and my supervisor, Professor Maria Musaji. Thank you.
Thank you, Vivian. Very clear, large patient list. One question from Julie. Why does surgery impact aniridic eyes more than those without?
Okay, and is that in terms of visual acuity? If so, I think patients who required surgery were those who
would then have had things like glaucoma, cataract, aniridia-related keratopathy. And so there may be more contributory factors which may be affecting their visual acuity to begin with.
Yeah.
And so in terms of comparing those who may not need surgery, i.e. they have milder forms of aniridia-related keratopathy, glaucoma or cataracts, I suppose that's the main reason why they may maintain a better visual acuity, because clinically they're less affected compared to someone who requires surgery.
Yep. And one of the things you didn't have time to cover is you had quite a lot of familial cases, intrafamilial variation. So, and also when you group patients with the same, well, let's just say stick to the familial cases. How much variation is there within families?
So we did do intrafamilial analysis, and actually there weren't too many variations. There was only slight variation in terms of iris hyperplasia recorded in 1 or 2 cases, but otherwise, I don't think we had any significant results of, a lot of variability.
Right. Good. I'm afraid we've only got time for a few questions, but thank you again for a very clear talk, and I'm sure there'll be more questions in the later period. Thank you.
Thank you.
Thank you, Viv. So moving on, we have next Alejandra Tamayo. She's a PhD student at the Institute of Research and Hospital Fundación Jiménez Díaz in Madrid, Spain. And she'll be presenting her work on functional characterization of non-canonical splice variants in aniridia by mini-genes and ex vivo approaches.
Good morning, my name is Alejandra Tamayo and I'm a PhD student from the Department of Genetics and Genomics at Fundación Jiménez Díaz University Hospital. I'm going to present part of my work, which is mainly focused on the functional characterization of non-canonical splice variants in aniridia by mini-genes and ex vivo approaches. Aniridia is a panocular rare disorder caused by defects on PAX6 gene. Using genomic-based tests, a pathogenic variant is detected in about 90% of patients. Thus, some of the patients carry variants of unknown significance located in non-canonical splice sites. Functional assessment of these variants is essential to reveal their implication in the pathogenicity of aniridia, because they could potentially interfere with exon intron recognition and cause an aberrant splicing process. So the aim of this work is to carry out functional studies of the impact on splicing of non-canonical splice variants by mini-genes and lymphoblastoid cell lines. So how we develop these gene molecular tools? Firstly, to establish LCS, lymphocytes of aniridia patients and control individuals are infected by Epstein-Barr virus, and the cDNA is studied by semi-quantitative PCR and Sanger sequencing. Secondly, to develop mini-genes, we clone the region of interest of PAX6 into an exon-trapping expression vector. Today we have developed 4 different mini-genes. These mini-genes are composed by 5' UTR, exons 5 alternative and 6, exons 5 to 7, and exons 8 to 11 of PAX6, respectively. Each variant is introduced by directed mutagenesis PCR, and finally, wild-type and mutated constructs are transfected into cell lines, and the splicing analysis is made as the same way as I've mentioned previously. By these molecular approaches, to date, a total of 25 variants have been studied or are now being studied by mini-genes, and 4 variants by LCLs. Today, I'll show you the functional characterization of one of these variants found in a patient of our cohort and how these molecular approaches could help in pathogenic classification. The variant under study is C.357 plus 6T2G. This variant, located at intron 6 of PAX6, was detected by NGS and classified as variant of unknown significance. This variant was present in the proband and his father of this family. Intrafamilial variable expressivity is observed in these two carrier patients. The proband presents complete aneuploidia, meanwhile his father suffers from iris atrophy and coloboma. So, in order to prove the implication of this variant in aneuploidia, a study with many genes was performed. Comparing the variant against wild type, this study revealed a complex pattern which includes 4 aberrant isoforms generated by the use of cryptic splice sites. We could also study the LCLs of the patient, and the results we obtained correlate with those observed with the minigene. So these two molecular approaches led a reclassification of this variant from variant of unknown significance to disease-causing variant. To sum up, our work confirms the involvement in aberrant splicing of C.357 plus 6 T2G variant using in vitro and ex vivo assays. This variant, previously classified as variant of unknown significance, was reclassified as disease-causing variant, allowing a more accurate genetic diagnosis and counseling. Many gene assays, including several exons and LCS studies, are useful strategies to deepen into the pathogenicity of non-canonical splice PAX6 variants. Thank you very much for listening, the organization for inviting me, and everyone involved in this work.
Well, well, thank you, Alejandra, for a great talk. One of the two questions really. The first is how robust are these assays? I mean, do they work well each time, or are the experiments very difficult to do?
Yeah, these assays are not easy to do because they have a lot of steps along on the process. But finally, we have set up this method, and it is very useful to deepen into the pathogenicity of these variants. And we have a background of using this method, so finally it's easy to do it.
In your hands, it's pretty robust. So the next, I mean, this is a question from the clinical point of view, in the sense that clinicians get a lot of, not just with PAX6, with other genes, variants of unknown significance, and it results in patients. And this is a very interesting way of trying to look at pathogenicity. But how much work does it take to do each mini-gene assay? So is there any way you can see in the future of speeding this up to be more clinically useful?
Well, more or less to do a mini-gene, maybe 2 months, 3 if all things are going well. So maybe if we have to study all variants of unknown significance we saw in patients, maybe it's not a useful tool if you want to have results in a short time. So it's not at the moment practical for widespread use, is the answer, I think. Yes. No, in my opinion, no, for the moment, because as I have mentioned, maybe if you are doing well, at least 3 months or 4, it's. Yeah.
Well, very interesting talk again. Thank you very much.
Pleasure. Thank you.
Thank you, Alejandra. The next speaker is Vishal Vora. He's a cornea and external eye disease fellow at the Department of Ophthalmology at Royal Victoria Infirmary in Newcastle. He'll be presenting aniridia-associated keratopathy, a retrospective analysis.
This is Dr. Vishal Vora. I currently work as a cornea and external eye disease fellow at Royal Victoria Infirmary, Newcastle. I would like to thank the organizers and my co-authors for giving me the opportunity to present this study. Aniridia-associated keratopathy is a chronic ocular surface problem of varying severity seen secondary to underlying limbal stem cell deficiency in up to 90% of the patients with aniridia. Besides glaucoma, it is the second most common cause of progressive vision loss in these patients, the underlying pathology being limbal stem cell deficiency. It can present as chronic corneal ulcers and recurrent corneal erosions. Chronic pain can be disabling in these patients. The main investigations for diagnosis of this condition include impression cytology and in vivo confocal microscopy. The aim of our study was to analyze the incidence of aniridia-associated keratopathy, visual impairment status, and associated ocular comorbidities in patients of aniridia, for which we retrospectively analyzed records of 30 patients being managed in ophthalmology department at Royal Victoria Infirmary. Also, we looked at the demographics, best-corrected visual acuity, aniridia-associated keratopathy grading, follow-up duration in these patients. The aniridia-associated keratopathy was graded according to the criteria proposed by Lopez and Garcia et al., with grade 3 being the most severe and 1 being the early stages of limbal stem cell failure. So coming on to the results, a total of 59 eyes of 30 patients were analyzed in the study, with the mean age at presentation being 13.3 years. The mean age at longest duration of follow-up was 27.9 years. 44% of the eyes were noted to have partial or total limbal stem cell deficiency based on slit lamp examination. Total medication burden noted in these eyes was 69% of the eyes being given glaucoma medication, 33% eyes being given lubricant preservative-free eye drops, 10% of the eyes being given serum eye drops, and 10% eyes being given low-dose continuous topical steroids. The surgical interventions done for these patients included bilateral cataract surgery in 11 patients, bilateral tube surgeries in 4 patients, unilateral tube surgeries in 5 patients, and unilateral retinal detachment surgeries in 3 patients. Coming on to the aniridia-associated keratopathy staging in eyes with limbal stem cell deficiency in this study, 46% of the eyes were graded as stage 1, 23% were graded as stage 2, and 31% of the eyes were graded as stage 3. Also, the vision was significantly worse in eyes with aniridia-associated keratopathy. 24 patients were registered as sight impaired, and of these 24 patients, 18 were registered as severely sight impaired. Associated ocular comorbidities in the study included 100% of the eyes with nystagmus, 69% of the eyes with glaucoma, and 42% of the eyes with cataract. So to conclude, the best corrected visual outcomes are worse in patients with aniridia-associated keratopathy, and also this contributes significantly to long-term visual prognosis combined with other comorbidities like glaucoma, cataract, and nystagmus. The lower incidence of 44% aniridia-associated keratopathy in our study can be attributed to lower mean age of the study population. Thank you.
Thank you again for a very interesting talk. I'll start with a question from Julie about why are some patients more prone to the keratopathy than others with
Thanks, Tony, for the question. I feel the reason for that could be underlying genetics. We still are looking at the genetics of these patients. We have only data for 33% of the patients in this cohort, but I think that is where the answer lies. Also, I think these patients come at a pediatric age when they come into the clinics, and they are being managed by pediatric ophthalmologists. I think they need some specialist intervention at early stages with drops like serum drops in early part of the disease to prevent any further progression of limbal stem cell failure, which might help with better visual outcomes later on.
Right. The question I have related to this is that did you measure tear production in your patients? Because one of the things I was not aware of for a long time was that patients with aniridia have very poor tear production, and I wonder whether that has any influence on the severity of the corneal disease.
Unfortunately not, because this was a retrospective study. We were looking at the records of the patient, and most of them didn't have a record of any Schirmers done for these patients. But from now on, we are definitely making sure any patient coming to cornea subspecialty, we do record the Schirmer's test, and it's definitely low, less than 5 in most of the cases.
Yeah. Well, thank you again, and thank you. A very interesting talk. I'm sure there'll be more questions in the chat later. But again, thank you very much.
Thank you, Tony.
Okay, now to the last presenter, actually. Her name is Jovita Urbaite. She's a residential doctor at the Lithuanian University of Health Sciences Hospital Kauno Klinikos in Lithuania, and she'll be presenting the spectrum of PAX6 gene mutations and genotype-phenotype correlations in a single Lithuanian center experience.
Hello, everyone. My name is Jovita, and today I will be presenting the research that me and my colleagues carried out, which is called the Spectrum of PAX6 Gene Mutations and Genotype-Phenotype Correlation, Single Lithuanian Center Experience. In this study, me and my colleagues analyzed retrospective data from the Lithuanian University of Health Sciences Hospital Kauno Klinikos for patients who had molecular genetic testing for PAX6 gene, and likely pathogenic covariant of unknown significance was found. The study was conducted between the years of 2017 and 2020, and we analyzed not only genetic but also demographical and clinical data, which allowed us to evaluate patients' genotype and phenotype. A total of 15 patients with likely pathogenic variant of unknown significance was found, and a total of 7 unique PAX6 gene variants was found. The majority of patients, 93.3% of them, did have a PAX6 gene variant inherited from a parent, and only 1 patient had a novel PAX6 gene mutation. Mostly of the patients, 66.7% of them did have a premature termination codon-inducing PAX6 gene mutations, which leads to the production of shortened and not functional protein or even a loss of the protein. And 33.3% of patients had missense mutations, which leads to the production of full-length but impaired function protein. And missense mutations are usually associated with less severe phenotype. However, in our study, all of the patients with premature termination codon-inducing PAX6 gene mutations did have aniridia, while only 1 out of 5 patients with missense PAX6 gene mutations had a diagnosis of aniridia. Of course, patients with PAX6 gene mutations had not only diagnosis of aniridia, but also other eye anomalies as well. 80% of patients had refractive errors, 73.3% of patients had nystagmus, 66.6% of patients had cataracts, 60% of patients had glaucoma, 53.3% of patients did have keratopathy, and interestingly, all of the patients with corneal anomalies did have the diagnosis of aniridia as well. Foveal anomalies and posterior eye segment anomalies were diagnosed in 46.7% each. Other ocular phenotypes such as strabismus, ptosis, or ectopia lentis were found in lower frequency. 60% of patients with PAX6 gene mutations had not only ocular but extraocular anomalies as well, with the most common one being neurodevelopmental and psychiatric disorders diagnosed in 20% of patients. 2 patients that have dysmorphic features, an example, dysmorphic ear cups and lower attachment of ears. 2 patients had neurological anomalies as well as hematological anomalies and obesity. Orthopedic anomalies, endocrine anomalies, and Sjogren syndrome was found as well. And 1 patient did have duplex kidney. So to conclude my short presentation, we did have found a wide variety of ocular and extraocular anomalies in patients with PAX6 gene mutations, from only mild iris anomalies to severe aniridia and extraocular phenotypes. and our study did confirm the connection between the type of PAX6 gene mutation and aniridia phenotype. Patients with premature termination codon-inducing PAX6 gene mutations are more likely to have aniridia than patients with missense PAX6 gene mutations. So thank you for your attention, and that is it for me.
Well, thank you again, Jovita, for a great talk and interesting genotype-phenotype study. You mentioned that the protein termination patients were much more likely to have aniridia. But what about other aspects of the phenotype? Was there any difference between the missense mutations and the protein terminating mutations in terms of the cornea or other aspects of the condition?
Yeah, so the most difference was with aniridia, as premature termination codon patients had mostly aniridia, but one patient with missense mutations did have Peters anomaly, which other patients did not have. And there were other eye anomalies like stationary night blindness with missense mutations, probably which are not connected with PAX6 gene mutations, but cataracts, glaucoma, and other anomalies were found equally in missense and premature termination codon-inducing mutations.
Well, thank you. And a question from Julie. This is a general question moving forward. We've heard a number of talks today about genotype-phenotype data, including your own excellent talk. How do we use all that data going forward for the benefit of patients? What do you think the benefit of these studies are, apart from this interest in science?
Well, I think it is easier when you know what phenotype might be in the future to warn the patients to choose the treatment and to diagnose aniridia and to suggest genetic testing for patients who had not had genetic testing and for their family. So it's one of the criteria, I guess.
Yeah.
And yeah.
Good. Well, thanks again for a very clear and interesting talk. I think there'll be other questions that are coming up in the chat, which will be put to you later in the chat rooms. So thank you again.
Okay. Thank you. Bye.
Julie Daniels, first of all, apologizes for difficulty she had. She had a very poor internet connection this morning, but she was able to see the slides and read the abstracts. First of all, I want to congratulate all of the speakers for doing an amazing job of putting across their science and results in the 4 minutes and doing it so well. It's a very difficult decision for the judges. Basically, the talks divided up into a number of very good genotype-phenotype papers, 2 studies on mini-gene preparations to look at the pathogenicity of rare variants, and a paper on the ICUP. And it was extremely difficult, but overall, we thought that the most complete and best study and best presentation was that of Hilda Pedersen on her very careful and difficult studies of OCT analysis of retinal structure in aniridia, and we were impressed by how she showed the changes in the outer retina and correlated it with mutation type and visual acuity. So again, it was a very difficult decision, but that was our final verdict. Thank you.
Thank you, Tony. So I think it's very brave and wonderful of everybody putting themselves forward to give these talks. It's incredibly good training, and in a way, it's a bit like tempering steel. You have the heat and the cold water, and it makes you stronger in the long run, whether you win or not. but actually, you know, it's such a difficult judging experience because everybody is very good. Very odd circumstances as well, having this online pre-recorded system with some people having difficulty in joining, like Julie. Anyway, it is really my great pleasure to give the Judges Prize to Hilda Pedersen, and the popular vote was for Philippa Harding to get the popular prize, which is donated by Aniridia Network. So I think that's a wonderful outcome, and the people who talked and did not win were fantastic too. And it really, I hope that you enjoyed the experience, and we'll carry on trying, and I'm sure you'll win as time goes on. Thank you very much.